the EMT theme, and now we have another very interesting talk from Marina Puchinskaya from Belarus, who will be giving a talk with the title Epithelial-mesenchymal Transition in Cancer Stem Cells. Same, similar, or different? Very good question. Let's see if she can answer it for us. So Marina, if you want to start sharing your screen.
Okay. Do you hear me well?
Yes, perfectly. Let's hope we can get your full presentation up.
I'll try. Okay.
Perfect. Good. Right. Go ahead. Thank you.
Thank you. It is always very difficult to talk about after such brilliant presentations as Mohit has done, but anyway, I have nothing to do. So my name is Marina Puchinskaya. I'm a clinician oncologist and pathology scientist from Minsk City Clinical Oncology Dispensary, Minsk, Belarus. And in my talk, I would like to continue the topic of EMT and cancer stem cells, about their possible interconnections and their possible influence on each other. So I have no disclosures. Epithelial-mesenchymal transition, as we have already heard, is a cellular process of transient acquisition of a number of morphological and functional characteristics of mesenchymal cells by epithelial cells. Functional characteristics mostly include motility and invasiveness that create pro-metastatic potential, and that is why EMT is considered to be one of the first steps for cancer cell metastasis. And morphological features include downregulation of epithelial markers and upregulation of mesenchymal markers. In our work, we mostly focus on ecadherin and encadherin, that is known as cadherin switch, though several other molecules like ecadherin and some else were also used in these studies. And the most important thing is that EMT is now widely considered a mechanism of treatment resistance to any known treatment strategies like radiotherapy, chemotherapy, hormonal treatment, targeted agents, and immunotherapy. So here we can see on this picture some of the inducers of EMT, like hypoxia and other changes in oxygen pressure, immune cells, mechanical stresses, changes in extracellular matrix, and more important, chemotherapeutics. It was shown that, for example, hormonal agents may also induce EMT in hormone-dependent tissues. And here you also see how many different markers can be used for EMT detection, and it again comes to a very good question by Professor Ben Baruch that really EMT is not the single state because very different markers may be expressed in those cells. So we know about that concept of hallmarks of cancer, and EMT is connected to practically all of them, like invasion, migration, analytics resistance, evasion of the immune system, and everything like that. And more importantly, self-renewal and multipotency, that are known characteristics of cancer stem cells. So first ideas of EMT and CSC interconnections came from 2009, work by Sandra Mani et al., who showed that induction of EMT in breast cancer cells also induced CSC phenotypes, CD44 high, CD24 low. And after that, a number of studies also shown that EMT and CSC phenotypes may be closely connected, and some scientists even consider them the same phenotype. It was thought that maybe those are like close processes or interconnected processes, or maybe even the same process. It is still unresolved question with many different ideas from various works. And what is known by now, for example, those are only like some citations that induction of EMT leads to CSC phenotype, again, money. Circulation tumor cells express both EMT and CSC markers, works from Casimir Bohr and several other teams. And this leads us to an important notion of the so-called migration cancer stem cell. And for example, evaluation of EMT and CSC markers in tumors may have prognostic value, an important clinical notion. So levels of possible EMT and CSC interactions are various, and practically at each level, those processes can be interconnected. So when we're talking about inducing factors, that is different changes that may stimulate both processes. It is known that, for example, oncogenic signaling, hypoxia, extracellular matrix, hormonal agents may stimulate both states. Several examples, for example, Jonas Fuchs et al. Showed that TGF-beta signaling, a well-known inducer of EMT, can also induce CSC phenotype due to activation of canonical SMART signaling in breast cancer models. Another important notion, as I mostly work on prostate cancer, so hormonal agents like androgen deprivation therapy in any technical type of castration may also cause EMT. It was a very important work by Sun et al. In 2012, who showed that ADT in vitro caused epithelial-mesenchymal transition in prostate cancer and higher aggressiveness and invasiveness of this tumor. However, it was also shown that cancer stem cells in prostate cancer are androgen receptor negative, and as so, they survive castration and they may be even stimulated or given growth advantage in the castration state. Regarding intracellular signaling and different signaling pathways that interact within the cell, again, the signaling pathways of EMT and CSC are closely interconnected, including signaling of WNT, beta-catenin, including PI3K, mTOR signaling, for example, already mentioned TGF-beta signaling. All of those signaling pathways were shown to stimulate both EMT and CSC phenotypes and characteristics. And the last level, that is genetic signatures. Again, we should note that both EMT and CSC phenotypes are not really stable and are not prescribed, let's say so, in genes. So we're talking maybe more about like any kind of epigenetic regulation. But however, some changes in gene expression of various genes may also cause and lead to both of these processes. For example, SLOC and ZEB proteins were both shown to induce EMT and CSC phenotypes in various experimental models. So again, what was already mentioned by Mohit, EMT and cancer stem cells are implicated in several important theories and important concepts that have changed the way we're thinking about cancer, metastasis, and treatment. So one of this is understanding of EMT as a metastable state. So we do not think about EMT like only epithelial state, only mesenchymal state, and just quick changes between them. So we now understand, including after Mohit's talk, that those conundrum between epithelial and mesenchymal cells, they are not really mutually exclusive states. So we have different values of epithelial and mesenchymal traits, and they can be changed. They can be changed due to different signals from microenvironment, including. And so we should understand that maybe for epithelial cells, maybe epithelial state is more stable, and we need some influxes, some stimuli to make epithelial cells change their state for mesenchymal one. So that is an important way we're thinking about cancer cells. So as a clinician, I can say that previously we were thinking that cancer cells are stable, bad but stable. Now we understand that they are bad and changing. So we're having not a stable target, but a moving target that makes the treatment even more difficult. Another important concept connected to CSC and EMT is the so-called migrating CSC concept. That means that when you have, for example, epithelial malignancy, epithelial cells, they may be changed into mesenchymal-like cells by EMT, and they, due to that, become more invasive and migratory. And they also get apoptosis resistance, that is very important for traveling in blood. And for example, when you want to get to another organ, it is very important not to die during your travel. But when we as pathologists look at metastasis, again, we see that most of them are really epithelial in nature, so they mostly resemble the primary tumor. This led for, in my opinion, a bit artifactual theory of mesenchymal epithelial transition. So we just had to explain that in any way. So we thought that maybe those mesenchymal phenotype can be again changed in the epithelial one in the metastatic niche. And this also led to the notion that maybe we need some stimuli to make epithelial cells more mesenchymal, and those stimuli are lacking in metastatic sites. But again, just pretend several cells coming into another organ, another microenvironment, another stimuli, another like molecules everywhere and everything like that. They need to possess some kind of stability and some kind of self-renewal possibilities just to give rise to metastatic lesions. So that made the concept of that possibly those cells that have come to the metastatic site, they possess some kind of CSC characteristics like self-renewal and differentiation ability, and only those cells that do possess those features can give rise to metastatic lesion. This concept is also experimentally proven by the fact that, for example, circulating tumor cells that are thought to be seed of those metastases, they often express both epithelial mesenchymal transition markers and cancer stem cell markers. There are a number of important works in this area, and this concept also changes the way clinicians are thinking about treating cancer, because now we understand that we need to treat not only stable cancer cells, but also that EMT and CSC phenotypes that are changing and that are mostly important for progression of the disease. Another important notion, for example, for me, it was very interesting that some markers that were thought to be classical EMT markers may also serve as CSC markers. For example, cell surface vimentin. Vimentin, as you know, is an intermediate filament protein that is highly expressed in the cytoplasm, and it is a classical mesenchymal, EMT marker, classical mesenchymal EMT marker. That is used in many works, but it was shown by several groups that it can also be present at cell surface in different cell cultures. And those cells with cell surface vimentin, they possess CSC properties. Interestingly, it was also shown that targeting cell surface vimentin augments cancer cell growth, and as so it can be an important therapeutic target. Those works are mostly experimental but they are thought to be very promising from the clinical standpoint. So by now we can talk about different similarities between EMT and CSC. It's like in childhood, find similarities. So both EMT and CSC are cancer mimickers of normal processes. We know that EMT was first described in 1955 by Elizabeth Hay as a developmental process, and after that it was shown to be a cancer metastasis and progression mechanism. Cancer stem cells are like cancer mimickers of normal stem cells, so that represent normal normal tissue hierarchy, and CSCs represent that in tumors. Another important notion is that both are phenotypes rather than cell subtypes. We should think of them as a number of characteristics, mostly functional and also morphological, but those are not stable phenotypes, stable subtypes of tumor cells that can be found and that can be, let's say, targeted. When we target some cells, some EMT cells or CSC cells, if they die, it was shown well in experimental models that other cells, non-EMT and non-CNC tumor cells, may then go to that state and develop more EMT and CSC cells. So killing those cells should be permanent, but not only one at a time. So again, both EMT and CSC are caused by complex gene expression and epigenetic regulations. So those they are not caused and they're not stopped by 1 or 2 or even 3 genes. Those are complex genetic programs that lead to those phenotypes. And again, though it's not completely agreed yet, but there are some nice works that have shown that any tumor cell may become EMT cell and CST cell. And those notion is very important again regarding treatment strategies. Both EMT and CST can be activated by oncogenic signaling pathways. That is understandable. So again, in the case of oncogene addiction, and those processes are not fully described by markers expression. We should, however, understand that expression of EMT markers and CST markers is a good way to find those cells, but if the cell has those markers, it doesn't mean that it is really CST or EMT cell. And again, as it was already noticed, there are very many markers for both processes, and we still do not know whether cells expressing different markers, they are similar in their functions and in their properties for cancer progression, or they are by yet different. So, regarding our own research, we are studying, unlike Mohit, we are studying human prostate cancer samples obtained during radical prostatectomies. We're using formalin fixed paraffin embedded tissues. Using immunohistochemistry and double fluorescence as most equivalent methods to study EMT and CST markers, and we are evaluating staining patterns, mostly semi-quantitatively, but we're trying to use quantitative methods and to develop them by now. So I would like to show you some of the results of our work, that is ectodermal expression patterns that were observed in our research. So membrane staining, that is quite obvious, and cytoplasmic staining, that is also seen in some cases and in some cells. It is usually less prominent than membranous one, and seen not in all cases. It is also well known that cytoplasmic ectodermal staining may be a marker of EMT. So again, this is how it can look like in various cases, and this is how encoderine looked like. In the same cases, it should be noted an important notion that ectodermal staining is mostly downregulated in all of the cells, and it is rather homogeneous in different cells. So in the same case, the level of downregulation is rather homogeneous, but for encoderine, the staining pattern is very unsimilar. Mostly, the expression is seen in some cells or in some glands, and or in some areas of glands, and usually we do not see uniform staining in all the cancer areas. So, encoderine is downregulated rather in all cells, but encoderine neo-expression is seen in only some cells, and we assume that maybe those cells that possess encoderine and also downregulate ectoderine are the most likely candidates to be really EMT cells. We evaluate ectoderine expression using the Image Scope software, and we used average staining intensity as a marker. It helped us to evaluate both number of positive pixels and the staining intensity. It was a very nice tool. And we, for example, using this tool could show that, for example, ectodermal staining is not related to the area of tumor, whether it is tumor center or more peripheral parts of the tumor nodule. It is not connected to Gleason score and the Gleason grade, and that is the level of tumor differentiation, and also it is not connected to clinical characteristics like TNM category or the presence of neoadjuvant androgen deprivation therapy. But for example, in the areas of most invasive cancers, ectoderine is downregulated, and it was shown with statistical significance. Regarding encoderine expression evaluation, we faced a problem of, unfortunately, false positive staining, like those Golgi pattern. We tried to use different methods, but we couldn't get rid of it, so we decided to assess the expression semi-quantitatively. And it was also shown that this expression is not connected to differentiation and TNM categories. And unfortunately, by now, we have studied only 50 cases, but by now we didn't find statistically significant correlation between N-cadherin and E-cadherin expression. Maybe due to a small sample size, maybe due to different patterns of staining, but by now the result is as is. Again, now we're trying to use double immunofluorescence, and we're trying to develop this method and use it in our own research, just to try to evaluate the co-expression of N-cadherin and E-cadherin in the same cell. So here you see in green E-cadherin stain and in red E-cadherin. And obviously in this cell, co-expression is seen, and so these particular cells, they are very scarce. As they only seen, maybe those cells can be really considered to be EMT cells. So again, environmental staining regarding possible link to cancer stem cells. So environmental staining was seen in normal non-cancerous glands at the level of basolateral membrane. Membranous cytoplasmic staining was seen in many cases. It's a normal pattern of staining for vimentin. But membranous staining was also seen in some cases. It was less prominent than cytoplasmic one, and it was also seen on the membrane regardless its area, so basolateral and apical regions. And again, if cytoplasmic staining was mostly widely seen, like E-cadherin, membranous vimentin expression was seen in only some glands and in only some cells within the gland. So we hypothesized that maybe those cells having membranous vimentin as a surrogate, possible surrogate of cell surface vimentin seen in experimental conditions, maybe those cells are candidates to be not only EMT cells, but cancer stem cells. And in future, we are also planning to assess the possible link between vimentin staining and cadherin staining and staining of per se CSC markers like CD44. I have shown those pictures already yesterday, and it's again fluorescence of CD44, CD133, and CD133 as conventional CSC marker. So to conclude, I can say that experimental evidence by now suggests that EMT and CNC phenotypes, they are interconnected and they can be induced simultaneously. However, we still have works that show that those states may be mutually exclusive, and in some works it was shown that EMT induction downregulates CSC features, so we can't say that those phenotypes are the same, but at the same time we can't say that they're different, absolutely different. But most of the results regarding those questions are obtained in experimental conditions. And we do not still know how to address this question in clinical samples and in patients. So this is a very interesting area of research. We are planning to conduct further research in this area, but it is still very, very interesting whether those experimental notions will really be confirmed in human samples. Also, it was shown that some molecules myself as both EMT and CSC markers like cell surface vimentin, and if yes, if we can hit both processes, EMT and CSC, by one target, it is a very good idea for a clinician because we can just try to develop some drugs to this molecule to hit both targets. And by now there are some antibodies targeting vimentin, but they're used unfortunately only in experimental conditions. But again, very, very prospective notion for clinics. And of course, more research is needed to understand the mechanisms underlying both of the processes and how those processes are interconnected. And if we do understand cancer, we can really treat cancer. So with that, I would like to thank to all the patients who gave their permission to use their tissues, to all collaborators, Belarusian State Medical University and International Sakharov Environmental Institute of Belarusian State University, and our main founder, Belarusian Republican Foundation for Fundamental Research. And of course, I would like to thank all the audience for their attention, and Professor Spatova and all the organizing committee for the opportunity to speak at this important meeting Please stay safe, and I wish all the best to you. Many thanks.
Thank you. Thank you very much, Marina. We're short on time, but if anyone has we can probably take one question. If anyone wants to actually ask the question or send it to the chat. While we're waiting for a few seconds, I just want to thank you for a very nice overview. It's actually quite interesting that this cadherin switch, we also see it in pancreatic cancer. So it's actually something very interesting with the cadherin and cadherin, especially in cadherin, the differential expression is also seen in pancreatic cancer. So I think that's quite interesting. And if no one wants to ask a question right now, please also feel free to send Marina questions in via the chat. I will ask you one question, Marina, if you don't mind. Perhaps I'm thinking about it. You talk about these moving targets, which I think is a quite interesting way to think about EMT. Would you try, or do you think that we talked about microenvironment factors that might influence EMT? Do you think metabolism might also have an effect on EMT, the metabolic state of the cell?
Yes, I think, well, we are not addressing this question in our work personally, but from the literature perspective, yes, metabolic reprogramming can really influence on EMT. First of all, hypoxia may change tumor cells metabolism, and it is one of the mechanisms how microenvironment and metabolism can be useful for EMT. Another important notion is that glucose metabolism may also be important for EMT state, and it was shown regarding moving targets and treatment strategies that metformin, a well-known anti-diabetic drug that was used for many years to augment glucose metabolism in diabetes mellitus patients. It was shown in experimental conditions to downregulate both CSC signatures and CSC markers and also EMT signatures. So by now, from a clinician standpoint, it is the most, let's say, the best way of maybe how we can treat both of this processes. And as metformin changes metabolism, yes, metabolism can really influence EMT.
I agree 100%. So thank you very much, Marina. Also, again, I stress, feel free to